Hello everyone, welcome to this audio podcast, part of this series, the Dezrztorm Didactic. This is another RPSG and Robotics collaborative. It's another Storm Robotics production, and I thought in this audio podcast I'd highlight some aspects in and around transplant, neuroimmunobiology, the immune system, transplant rejection, and neuroendocrinology, and sort of provide a retrospective, and again, as in the audio podcast previously, as highlighted in the audio podcast previously, this series and this audio podcast as well is aligned to the International Pediatric Transplantation or Transplant Association, or IPTA 2023. And so to begin with, I thought I'd provide some aims around this work. The aims of this work, or this research, was to highlight has been to highlight how specific immunobiological and neuroendocrine markers and sort of neuroendocrine markers can reduce chemokine migration and acute allograft rejection, or acute allograft rejection, or AAR. So the mechanisms around this. In context of the mechanisms, the pre-transplant phase. The pre-transplant phase is just as important or central as the post-transplant phase or period, and it is postulated through this works and in as much as context around neuroendocrinology. For example, that Selye's general adaptation syndrome, which was derived in 1936, and specific neuroimmunobiology and neuroendocrinology should be considered and aligned to transplant immunobiology in context of allograft or transplant prognoses. So the background around this is that cytokines are small molecules that signal between cells, inducing growth, differentiation, chemotaxis, activation, enhanced cytotoxicity, and/or regulation of immunity. Chemokines are cytokines. In effect, chemokines are cytokines which direct cell migration and/or activate cells, and chemokines closely relate or are related to cytokines that are produced by many cell types in response to infection or physical damage. They activate or direct effector cells to sites or tissue damage, and/or regulate leukocyte migration or trafficking into tissues. So in context of immunobiology, more specifically, there's in context of immunobiology specifically, CC chemokines are chemotactic for monocytes, and then CXC chemokines are chemotactic for polymorphonuclear neutrophils, or PMNs. And chemokine receptors are expressed on specific cell populations, permitting different chemokines to have selective immunobiological activity. Chemokines are primarily involved in attraction of leukocytes, so for example, lymphocytes, monocytes, and neutrophils, and they can be released by many different cell types, and serve to guide cells involved in innate immunity and lymphocytes in adaptive immunity. Some chemokines also have roles in the development of lymphocytes, a migration and angiogenesis. Where angiogenesis is the growth of new blood vessels. It is evident through literature and sort of retrospectively that cytokine molecules are, are, um, are potent molecules for activating and directing effector cell populations to the site of damage, as well as for controlling leukocyte migration into tissues. So now, to attain a long term transplant prognoses or survival has been perhaps the ultimate challenge for a variety of organ transplant disciplines or specialties, and immunosuppression or IST reduces the activation or efficacy of the immune system. Some portions of the immune system itself have immunosuppressive effects on other parts of the immune system, but clinically induced immunosuppression is carried out through pharmacotherapy. This is usually done to prevent the body from rejecting an organ transplant or for the treatment of specific autoimmune diseases. So, for example, systemic lupus erythematosus, for example, or SLE. When an organ is transplanted, the immune system the immune system of the recipient will sort of recognize or knows it as a sort of a foreign tissue, and it prompted to attack it. And cortisone, in context around this, cortisone was perhaps one of the first immunosuppressants identified. The more effective azathioprine, the more effective immunosuppression referred to or known as azathioprine was identified in 1959, but it was not until the discovery of cyclosporin A in the 1970s that trans that in aligned to transplant surgery found sufficiently powerful immunosuppressive. Now, um, other components, other pharmacotherapies also include monoclonal antibodies or MABs, which are antibodies that are identical to the immune system, because or have been identified to be close to the identical to the immune system, because they are produced by one type of immune cell, and all clones of single parent cells. Monoclonal antibodies, or again MABs, have advanced from involving fusion of immortal cell, a myeloma tumor cell, with a predetermined antibody producing B cell. The fusion is done by ensuring cell membranes are more permeable. Initially, mouse monoclonal antibodies, for example, were not ideal pharmacotherapeutics, because differences between human and murine genetics. This has been dealt by humanizing murine antibodies, or by making fully human monoclonal antibodies. It has now become standard to use monoclonal antibodies with immunosuppression, but also as immunosuppression or IST. Now, The question now the question then becomes is, how does neuroimmunobiology and neuroendocrinology associate? So stress is in around this stress is a medical term or terminology for a broad range of strong external stimuli, so both physiological and psychological, which can cause a physiological response called a physiological response called the general adaptation syndrome, or linked to the general adaptation syndrome, again first described in 1936 by an endocrinologist, Hans Selye, in the journal Nature. Selye described the general adaptation syndrome as having three main sort of aspects. Number one, alarm reaction, where the body detects external stimuli. Number two, adaptation, where the body interacts defensive countermeasures against a stressor. And three, exhaustion, where physiologically the body runs out of defenses. So it would seem acceptable that stage. So in line to sort of neuroimmunobiology and neuroendocrinology, it would seem acceptable that stage two, again where adaptation, in context of adaptation, where the body interacts defensive countermeasures against a stressor, that stage two corresponds to um, that corresponds to episodes of acute allograft rejection, or at least in by way of a sub question, around this, where studies of the immune system, part of the question, and then where study on the immune system and psychology become a direction which is significant and clinically important, or have been clinically important. So a change in the amount of natural killer cells, NK cells, and stress influence activity is an important defense mechanism of the body, and it constitutes a component of the preparation for defense against potential pathogen penetration. So when individuals or people get angry, for example, the entire body responds. It instantaneously becomes tense, and physiologically and automatically the body moves toward fight or flight. And then physiologically the entire body becomes relaxed. Stress hormones in this context can also change the immune cell behavior on the activity of the entire immune system. So there are studies and investigations involving transplant. Studies involving transplant immunobiology and neuroimmunobiology should be included in investigations to better understand, for example, acute allograft rejection for those recipients where transplantation has taken place. And thus to advance areas of advanced aspects of clinical medicine in and around this and around organ transplantation. Now, in line to this, there's original research by Rossi and Zlotnick, from in early 2000, in the 2000 duration, where they quite nicely put had developed a publication that looked at the biology of chemokines and receptors. Now, from a broad crux basis, biology of chemokines and receptors have several where chemokines have several functions, or have several bases where they are important. So, for example, chemokines are important for T helper cell type 1 and T helper cell type 2 development, angiogenesis and angiostasis, metastases, cell recruitment, inflammation, lymphoid organ development, lymphoid trafficking, and wound healing. So in the original works by Rossi and Zlotnick, there's an illustration, if you like, in around the importance of the chemokine and the receptor and receptors, and their importance in a variety of immunological functions. I'm not going to sort of highlight a little bit around this works in context of the literature, in context of a retrospective on the literature. So it was for example, to begin with, in around this, O'Leary in 1990 highlight that chronic stress has been associated with the stress with suppression of immune function, and there is evidence that the immune system may not adapt over time. It was Adder and Cohen in 1993 that highlight both conditioned and stressful stimulation exert biologically effects that can alter development and or progression of immunological mediated pathophysiological responses. It was Dabar and team in 1995 who informed that significant and selective changes in numbers or percentages are identified in peripheral blood leukocyte during stress in subpopulations of laboratory rat models. And it was Evans and team in 1995 who examined how stressful events and depression may affect key cellular immunological parameters in subjects with and without HIV infection, and cytotoxic T cell lymphocyte phenotypes. It was Mills and team in 1996 who examined the relationship between psychological characteristics and immune responses on acute laboratory stressor, and such distribution may significantly affect the ability of the immune system to respond to potential or ongoing immune challenge. It was Shedlovsky and Schmidt in 1996 who discussed in their work the stress and the immune system, and how acute stresses and physical activity have been shown to transiently enhance immune responses, and these stress effects on the immune system to be the stress effects on the immune system on immunity seem to be mediated through endocrine factors, since hormones, neurotransmitters and neuropeptides can interact with cellular components of the immune system. It was Goebel and Mills in 2000 who examined the effects of acute psychological stress and exhaustive physical activity on the expression and density of adhesion components referred to as adhesion molecules. So, for example, selecting LFA type 1 and intracellular adhesion molecules, ICAM type 1, on monocytes, granulocytes, and lymphocytes. Segreger and colleagues in 2000 who highlighted uh, who uh, who highlighted that chemokine receptors are present in acute allograft rejection and correlate with distribution of T cells. It was Hancock and team in 2002 who highlight varied chemokine receptors in acute allograft rejection. Segreger and colleagues in 2000 who highlight chemokine receptor. Hancock, Segreger and colleagues in 2000 highlight chemokine receptors are present in acute allograft rejection and correlate with distribution of T cells. It was Hancock and team in 2002 who highlight varied chemokine receptor expression in cardiac allograft rejection. It was Cockwell and team in 2002 who demonstrate the importance of the role of chemokines and chemokine receptors in acute allograft rejection. It was Lawson and team in 2001 who informed acute stress may signal an increase in at least some aspects of the cell-mediated or T helper cells or Th type 1 Th type 1 driven immune response. It was Maddock and Pariante in 2001 who also say that chronic stress appears to result in suppression of the immune response, whereas activation and suppression have been associated with acute stress. And Sternberg in 2001 who informed physiological levels of glucocorticoids are immunomodulatory rather than solely immunosuppressive, causing a shift of cytokine production from primarily a pro-inflammatory to anti-inflammatory pattern. And such patterns of cytokine production can be characterized as Th type 1 or Th type 2 and Th2 or Th type 2 and roughly correspond to cellular or humoral immune responses. It was works by Redwine and colleagues in 2000. Redwine and colleagues in 2003 who hypothesized that activation of a psychological stress response psychological stress response increases autonomic activity enhances immune function by inducing significant increase in numbers of leukocytes at sites of inflammation. It was also Redwin and team in 2003 who further discuss in their work or highlight also in their work that few human studies and investigations have examined the effects of stress on leukocyte motility or leukocyte motility, and to date there have been no sort of published works examining psychological stress and leukocyte or lymphocyte chemotaxis. Chemotaxis did not in works chemotaxis did not chemotaxis did not correlate or has not correlated with catecholamine levels, suggesting that another mechanism of activation may also exist, which increases chemotaxis after stress. It was Yoon and team in 2004 that demonstrate that chemokine receptors play significant roles in the development of chronic rejection and may serve as potential therapeutic targets for allograft made them serve as potential therapeutic targets for cardiac allograft vasculopathy. And it was Jiafeng and team in 2005 who demonstrate that the unique role of chemokine molecules in the maintenance of cardiac allograft tolerance mediated by natural killer T cells or NK cells or NKT cells in a laboratory murine model or mouse model, and it was Ulig and Cullis in 2005 that informed that there are or is plurality of how neuroendocrinological factors can affect and might influence immunity and immune system mediated disease. There are consistent and convincing reports of links between stress and disease onset and progression, and so prospectively a perfect model in context of I'll say here what's referred to as tolerance, for example, would be where patients are stress free before transplantation. For example, before organ transplantation is done or considered, and in context of therapy is also referred to, for example, replacement therapies and or prophylaxis, and throughout time awaiting an organ transplant, and this may well suppress those chemokine receptors that lead to acute allograft rejection episodes, sort of post transplantation or post transplant. In original works by several authors, there is there's works there's reference made to how certain chemokines are linked with particular findings in context of the immune system, and so for example it was chemokines referred to as rantes interleukin 8 and CXCL8, ENA78 and CXCL5. The findings around these particular chemokines, the findings that highlight in works by Patterson and team in '94, Mutabarak in '94, Mutabarak and team in '94, and Schmoder and team in '95. Highlight that express that findings around these particular chemokines expressed by endothelium and infiltrating cells, infiltrating cells in line to acute allograft rejection, and there's increased levels in urine and in context of acute allograft rejection, and mRNA increased increased expression in aligned to acute allograft rejection versus non-acute allograft rejection. And in original works by Inston and Cockwell in 2002, works referred to as chemokine and receptors identified in renal acute allograft rejection. There is sort of an overview, if you like, in and around chemokine and receptors identified in human renal acute allograft rejection, and where again different chemokines and the findings and the sort of and where investigators retrospectively have identified specific laboratory findings around these particular chemokines, and how they're important in aligned to in aligned to organ transplantation. And so a sort of from a perspective view where prospectively where investigations where further investigations can be could be conducted, for example, or could be carried out. I'm going to highlight here now some context around that around this. So chemotaxis is a methodology to investigate transport or migration towards a concentration gradient of chemoattractant for leukocytes. So for example, again, neutrophils, monocytes, and lymphocytes, or other migratory cells, and so an upper chamber containing a suspension of cells is separated by a membrane from a lower chamber containing medium with chemoattractant. Chemotaxis of the cells from upper chamber into lower chamber occurs over a predetermined duration. The cells in the bottom chamber may be counted directly. So, for example, when using a homogeneous population of cells, or may be stained for defined markers. So, when, for example, when assaying a mixed population of cells and analyzed through or by flow cytometry, where flow cytometry is used in the analysis of different cell types to sort of understand where physiologically or where physiologically there can be certain cells causing where certain cells can cause can cause problems, for example, physiologically or immunologically, and so in aligned to this, using a T cell system in isolation, T cell culture system in isolation would be helpful to observe chemotaxis as follows. So, for example, to observe chemotaxis for chemokine receptors, cellular adhesion molecules, and MABs, and thus use flow cytometry analysis to identify movement of particular cell types. In this context, measuring stress neuroendocrine levels pre and post transplant could be helpful, and complementary investigations. Monitoring stress levels in pre transplant duration is and would be of importance. A study with chemotaxis assays could potentially observe catecholamines with repeated stress, and this potentially would allow to help identify which hormones influence chemokine receptor migration. A study that incorporates chemotaxis and monoclonal antibodies with neuroendocrine post-transplantation would also be complementary investigation. Comparing the pre-transplant phase and observing chemokine receptors and neuroendocrine a neuroendocrine with a monoclonal antibody or MAB therapy would be important. And with this, examining the effects of acute stress and exhaustive physical activity on chemokine receptor sort of migration would also be a sort of a complementary sort of investigation. Uh, Studies with natural killer cell or NK cell activity and cortisol with chemokine receptors in pre and post transplant phases may also be important. Conducting investigations that relate acute stress or chronic stress, cytokine activity, and depression may show significant correlations between the immune system and neuroimmunobiology. Measuring neuroendocrine function with hypothalamic adrenal axis or HPA and chemokine receptor migration in pre and post transplanted recipients could be and would be important. So now with this, I'm now gonna highlight some conclusions, some sort of overall conclusions around this work surrounding transplant neuroimmunobiology in the immune system, transplant rejection, and neuroendocrinology. Um, More work is required linking immunosuppression or IST, monoclonal antibody therapy, and cell surface targets and neuroimmunobiology with neuroendocrinology. The immune system has generally has a suppressed action post transplant, i.e., to reduce leukocyte proliferation, which would normally act against a stressor, i.e., organ transplant. In this case, organ transplant. This works, or this work, the aim of this works was to establish and put forward a sort of an overview surrounding organ transplantation, neuroimmunobiology with neuroendocrinology in context of transplant immunobiology from a laboratory perspective. And whilst literature informs that chemokines surrounding transplantation provide a basis, it is now anticipated, or should be at least anticipated, that this work could be used as a guide to prompt procedures prospectively across different specialties, and using a multidisciplinary research effort should enable professionals to come together to achieve a better understanding of transplant tolerance. Based on Selye's work in 1936 and the general adaptation syndrome, or GAS, and stage 2, where the body engages defensive countermeasures against a stressor, it is proposed that chemokines appear to be integral players in this complex and dynamic process. A combination of neuroimmunobiology with neuroendocrinology in context of transplant immunobiology from a laboratory perspective to understand transplant immunobiology, and in context of stress, and Selye's work is still important. Um, and uh, yes, aligned to this, again, as in the case with the previous audio podcast, the in online there will be some citations or references that viewers can check out at their own time by putting the information by putting the citations into search engines, for example, online, and where those listening or watching can check out at their own time. And so with that, I'm gonna sort of end this audio podcast in and around a transplant and neuroimmunobiology, the immune system, and transplant, rejection, and neuroendocrinology, where this was a sort of a retrospective, and so with that, I'll say thank you for tuning in once again. This has been an RPSG and Lobostics collaborative. This has been the Shientist on this series, this audio podcast series around the Dezrztorm Didactic. This has been another Storm Lobostics production. Thank you for tuning in.